Okay, so 7.4 is where we left off. So if we recall that we were talking about diffusion, and we talked about passive transport and all the things facilitated diffusion, things moving from high to low concentration or down their concentration gradient. It doesn't require energy. None of the osmosis, diffusion, anything moving from high to low concentration requires no energy, and that's why it's called passive transport. So this next section is about how we get things across the membrane through a process called active transport. And active transport means it requires energy, like when you are active, you require energy. So we talked about facilitated diffusion. That, remember, what facilitates diffusion? Proteins. So proteins help with things getting in and out of the cell. That's like this picture up here. It's passive because the solutes move down their concentration gradient. What does down their concentration gradient mean? High to low concentration. But there are some other types of transport proteins that can move solutes, what we call against their concentration gradient, or up the concentration gradient. What does that mean? It means going opposite of what naturally happens. So against or up the concentration gradient means low to high concentration. And that requires energy. So that's the title of the next thing. So active transport moves substances against their concentration gradient, low to high, requires energy, and the form of energy is in that energy molecule ATP. Remember, has that phosphate group, which was one of our functional groups, has 3 of them, and when they come off, it releases lots of energy that can do cellular work, like move things against the concentration gradient. Active transport is performed by specific proteins. So specific proteins, so this is referring to the shape. So certain shape proteins allow certain things to go in and against the concentration gradient and move either into or out of the cell. So an example of one of these proteins that does this is called the sodium potassium pump. Whenever you hear the word pump, this means it requires energy. So never heard of something before and it's pumped. Oh, okay, it requires energy, and then that usually means it's going against the concentration gradient or low to high. All right, so the next picture shows you what goes on in the sodium potassium pump. So the pictures here are numbered 1, 2, 3, and then we go over here to 4, 5, 6. So this kind of happens in a cycle, and actually then after 6 we start back again at number 1, and so this can be a cycle there. And so let's be clear about what we're looking at here. Here's a protein that has a certain shape, and notice it has these binding sites that will bind certain things to it. In this case, it's going to be sodium ions. This says the extracellular fluid, so this is outside the cell. Here's your lipid bilayer, and this says cytoplasm, so this is inside your cell. And so if you notice here, the sodium binds to these specific spots, these binding sites on the protein. And once it does it, notice that there's 3 binding sites. So then you have all 3 binding sites filled with the sodium. Notice here ATP. ATP is going to be used, that's our energy. ATP, when we use ATP for energy, a phosphate group comes off. So it becomes ADP plus, whoops, that's supposed to be plus phosphate. And so that's what happens here. That means that energy is used. And what is that energy used for? The shape change of your protein. So notice here it says phosphorylation causes the protein to change conformation. Phosphorylation means that the phosphate group that was once part of ATP is now bound to this protein here, and it facilitates that shape change, which then, notice that the binding sites change shape and the sodiums go out. So therefore we have 3 Na pluses going out of the cell. All right, so now notice that the shape change for this other triangle binding site has changed as well. So now if we go to picture number 4, another ion called the potassium ion binds to those two sites, and there's only two sites for that. So the potassium binds, and then if we go from this picture to this picture, look at here, the phosphate group is no longer attached to that. That loss of that phosphate group makes and causes the protein to go back to its original shape. So it's back here in its original shape, and the potassiums then leave. Notice here, from here to here, as it goes back to the shape, the binding sites from here, this picture to this picture, they've changed shapes, and so the potassiums go into the cell. So now we have two K pluses that go into the cell. So let's draw here. So if this is a cell, what's going on here is the sodium-potassium pump is a protein in the cell, so this is the pump. And so what does it do? It pumps 3 sodiums out and pumps two potassiums in. And so that's kind of what it does. Now, and they're going against the concentration gradient, then, which would mean then that your lower sodium concentration is in the inside, higher sodium concentration on the outside. And that's why sodium is going from low to high, which is why it requires ATP to pump it against its concentration gradient. Potassium then is being pumped in, which means then the low potassium concentration is on the outside of the cell, and the high potassium concentration is on the inside of the cell. So it is going from low to high, therefore requiring energy. And so that's what the sodium-potassium pump does. Now, if we look at the charges of this, notice here the cell is losing 3 positive charges and only gaining 2 positive charges in return. So therefore, if you're losing 3 positives and only getting back 2 positives, what happens is as the sodium-potassium pump occurs, the cell becomes more negatively charged. So in here, the cell has a slightly negative charge. So I'm going to write that here. So more positives leaving than entering equals the cell becomes negative compared to where? Compared to the outside solution. Okay? And so we're going to see that this becomes important in getting ions in or out of the cell. And it'll become more important also as we get into and talk about how your nervous system works and neurons and so on. So I'm going to introduce it to you now, and then we'll come back to it also a little bit later. All right, so the inside, and this is true for cells, the inside is more negative than the outside. All right, and so we have this charge difference across the membrane. They call that the membrane potential. All right, so that's active transport. So this next picture just gives you a overview of the types of passive and the types of active transport. So passive, this is simple diffusion. It just says diffusion here. We can write simple diffusion. This just means it goes through the lipid bilayer. So from high to low concentration, no energy required. Then we have facilitated diffusion. So remember we had channel proteins. This is a channel protein right here. Channel protein. And then this one was a carrier protein. And they're facilitated by proteins, and they're called diffusion because the molecules are moving from high to low concentration. Doesn't require energy at all. Active transport, the molecules are going from low to high concentration. It involves a specific protein, and ATP is involved. And so this picture here of the sodium-potassium pump. All right, so that's your active transport. So if we look at the next thing here, says maintenance of membrane potential by ion pumps. So let's talk about what membrane potential is. So membrane potential is the voltage difference across the membrane. So what does that mean? Voltages, we'll talk about that being the charge difference. So back up here when we talked about the sodium-potassium pump, the sodium-potassium pump, as it pumps these ions and creates this negative inside of the cell compared to the positive outside of the cell, the outside is more positive, that's what's called a membrane potential. We'd say that there's a voltage difference across the membrane, and this wouldn't occur without the pump. All right, the pump pumping the 3 positives out and 2 positives in. So we have this charge difference across the membrane. And so this plays a role in getting ions across the membrane. So 2 combined forces collectively called the electrochemical gradient. Electro from electricity, charge. Chemical meaning the concentration of the molecules. Drive the diffusion of ions across the membrane. What's an ion? It has a charge, an atom with a charge. It's either gained or lost electrons. And so it has a charge. So let's look at how that could play a role. So for instance, if you have a cell that is negative on the inside and you have positive ions here, and you have a particular protein that allows those positive ions to go in, those positive ions can go in because of one of 2 different things, because it goes from a high concentration to a low concentration. But another thing that might play a role is that opposite charges attract, so these positive ions are actually attracted to the negative inside of the cell. So when we're talking about ions, there's 2 kinds of things that kind of determine which direction it's going to move. The concentration gradient, but also the charge difference. And so that's what these 2 here explain. So the chemical force is the ion's concentration gradient. So the ion can move from high to low concentration. And then the electrical force is the effect of the membrane potential. That's the charge difference, remember? So it's the charge difference across the membrane. So for instance, let's write an example here. The negative inside attracts positive ions. And that's what I just drew in the picture on the previous page. So you have this negative inside the cell attracting positive ions. And so, in animal cells, we have the sodium-potassium pump that creates that negative inside. In other types of cells, there's another type of, it's called an electrogenic pump, is a transport protein that generates the voltage or charge difference across the membrane. And in plants, and fungi and bacteria, that main electrogenic pump is called the proton pump. In animals, let's write this, in animals, it is I'll just put the sodium-potassium pump, which we just talked about. So in animals that don't have in plants, fungi, and bacteria, they have this proton pump, which pumps hydrogen ions against the concentration gradient. So this pumps H plus against the concentration gradient. What does this mean? So in here, inside the cell, there's a low concentration, and outside the cell, a high concentration. And that's what they're trying to show you here with the ions here. And so it's using ATP, hence the pump. All right, remember, pump requires energy, pumping it against its concentration gradient. All right, and so, why would the a plant want to do this? All right, so why is it that they might want to pump H pluses out? And it's because sometimes these H pluses help with the movement of other things into the cell that the cell needs. And so in comes what we call co-transport. Co in front of something means that it happens together, they work together. So co-transport occurs when active transport of a solute indirectly drives the transport of another solute. So we're actively transporting the H pluses. And so then what's going to happen is it's creating a high H plus concentration. The H pluses will want to diffuse without the use of energy back into the cell. So plants commonly use this gradient of hydrogen ions, this high concentration inside the cell versus out, generated by this proton pump to drive the active transport of other nutrients into the cell. So that's what the next picture, if we flip it over, shows you. It shows you sucrose coming into the cell. So I'm going to put here an example. Sucrose would be an example of one of those nutrients. So you look at this picture here. This part right here was the same picture as before. So the H pluses pumped from low to high concentration. All right, so the cell is expending energy to do that. So now it's got a really high H plus concentration on the outside of the cell. So why would the cell expend energy to do that? Well, that energy helps to create that negatively charged inside the cell. But then what's going to happen is the H pluses are going to diffuse through another protein. Notice that there's a different protein embedded in the membrane. Notice it says sucrose H plus co-transporter. This protein transports both sucrose and hydrogen ions across the membrane. But the thing is, in order for sucrose to get in, you have to have H pluses diffuse as well. So therefore, it's worth it for the cell to expend energy to make a really high H plus concentration. So now the H pluses can bind here. So this H plus right here, let's get a marker here. This H plus is going to come over here and bind here. And then notice that there's a different binding site with a different shape. That binding site fits the shape of sucrose. And so sucrose is going to come in. And once these both bind, they're able to go both into the cell. So the H plus diffuses into the cell. Why diffusion? High to low. Sucrose goes from high to low. And so sucrose comes into the cell. And so it was worth it for the cell to expend energy to pump H pluses out, because then the H pluses are going to help sucrose get into the cell, and sucrose into the cell can be used for energy, and so on. And so therefore, that's really important for the cell. And so it's worth it to spend that energy to get that sucrose in. So this is another type right here, this part. So this part up here is active transport, and this is facilitating. The proteins are facilitating this process. And so that is an example of a useful expenditure of energy and things going from low to high, active transport, and helping the cell out and bringing in sucrose. So we've talked about passive transport, all the diffusions, active transport against the concentration gradient. And the last thing we've already kind of talked about, especially with chapter 6, is bulk transport across the membrane, and this is exo and endocytosis. So let's look at this. Small molecules in water enter and leave the cell through the lipid bilayer or through the transport proteins. That's how a lot of things get into and out of the cell. But if the molecule is super large and can't either go through a protein or go through the lipid bilayer, these large molecules, such as polysaccharides and proteins, can cross the membrane via vesicles in some cells. So exocytosis. Exo means exit. Cyto means the cell. So exit the cell. So the process of exiting the cell. So in exocytosis, transport vesicles migrate to the membrane. fuse with it, and release their contents. Many secretory cells use exocytosis to support their product, or to export their products. So I use the example, we talked about this in chapter 6, the endomembrane system. So remember the endomembrane system, you had the Raphier made proteins. They went into a transport vesicle, went to the Golgi apparatus, and the Golgi apparatus modified them and packaged them into yet a different transport vesicle. So now I'm going to draw this. So here's your transport vesicle with your completed protein. And so then where that transport vesicle went often is the cell membrane. So if this is the cell membrane, so all this happened in the cell, the transport vesicle comes over here, fuses with it. So as it fuses with it, it opens up like this, and that protein that was made is exported from the cell. And I use the example of the lining of your stomach releasing enzymes to digest your food in this manner. So this is exocytosis. On the flip side, to get large molecules into the cell, we have endocytosis, means into the cell. So in this case, the cell takes in macromolecules by forming vesicles from the plasma membrane. And I use this as an example when I talked about unicellular organisms taking in their food. This is the reversal of exocytosis. So here's, let's say, your unicellular organism, and here's the food particle. So the food comes over here, and then the membrane indents around that food particle, and then comes over here and pinches off until eventually you have, this would be a food vacuole with your with your food for this unicellular organism within there. All right, and so that's your food vacuole, and that would be endocytosis, bringing in large molecules that can't get through the lipid bilayer or a protein, and so they engulf them that way. There are 3 different types of molecules that they can bring in this way. So the first type of endocytosis is called phagocytosis. This is cellular eating, so this is taking in solids. So the particle it's bringing in is a solid particle. There's penocytosis, where it's taking in liquids, cell drinking, so it's taking in liquids. Always remember that Pinot is a type of wine, all right, so it's a drink. And so it's taking in liquids. So it's indenting and taking in the extracellular fluid and any molecules in the extracellular fluid. And then lastly, there's receptor-mediated endocytosis. This is this picture here. So this picture is showing you, notice that there are proteins on the membrane of the cell. And so it's indenting its membrane, but there are these proteins, and these ligands, these molecules can bind to these receptors right here, these receptors on the membrane. And so what happens is, as this indents, it waits until all these triangle-shaped molecules bind to it and then pinches off and brings those triangle-shaped molecules into the cell. So what this is it gives a way for the cell to take in things that are in kind of low concentration. So it waits until these receptors are bonded to these molecules and then takes them in. If they just indented randomly and tried to get them in, there might not be any in this particular spot, any of these molecules. So therefore, this ensures that you get molecules that the cell needs to take in by allowing them to bind to these receptors, and then you take in a higher quantity of those as that pinches off. So these guys here, you just need to be aware of this. I'm not testing on the names. So you just need to know that through endocytosis, they could take in solids, liquids, or go through this process, which is another type of endocytosis. So I'm not going to say penocytosis is, or you don't have to come up with that name. All right? And so that is the end of Chapter 7. All right? So we will continue with the lab tomorrow and wrap up on Thursday, and our test will be on Friday. Have a great night.